The article of choice is titled Interactions of Trithorax and Polycomb Corto enhancer with methylated ribosomal Corto.
Chromatin structure strongly impact on regulation of gene expression. Post-translational histone modifications called epigenetic marks are recognized by protein complexes that shape chromatin. Chromodomains were first identified in Polycomb and heterochromatin protein 1. They are found in many other chromatin-associated proteins that belong to three classes according to their global structure. The Corto chromodomains is a functional chromatin targeting module. Surprisingly, peptide pull-down mass spectrometry and Biocore show that the Corto chromodomain interacts with the nuclear ribosomal proteins and notably binds with high affinity methylated on lysine.
Drosophila melanogaster stocks and crosses were kept on standard medium at 25 centigrade. RPL12 mice transgenic lines were established by standard P-element mediated transformation. Overexpression was carried out using GAL4 drivers, either ubiquitous or wing-specific. 5 females bearing the GAL4 driver were crossed with 3 males bearing the UAS transgene as a control. Crosses were transferred to new vials every third day. The GAL4 and RPL12 mice lines were isogenized for 6 rounds with the isogenic lethality was calculated as described: polyacrylamide gel and stained either with blue or with silver quist staining kit. Bind were excised from the gel and analyzed by LSMS mass spectrometry. Cytoplasm and nuclear extract were prepared from 0 to 16-hour embryos. GST Corto CD were covalently linked on agarose bed using the GST orientation kit. Following manufacturer's instruction, 1 mg of protein extract was incubated with 200 microgram of purified GST Corto CD in binding buffer 1 hour at 25 centigrade. After 5 washes in binding buffer, bound polypeptides were resolved on a large 15% SDS polyacrylamide gel and stained either with blue or with silver quist staining kit. Bands were excised from the gel and were analyzed LCMS mass spectrometry.
Transgenic flies, overexpressing Corto CD or Dautilus, were perfectly viable and had no visible phenotype. Overexpression of Corto using the same drivers was 100% lethal. Escapia flies displayed a rotated genitalia and duplicated macrochaetae as well as very penetrant homeotic phenotypes. Many flies presented a partial transformation of Arista into homeotic phenotype called Aristopedia that could reflect downregulation of spineless Aristopedia gene. Males overexpressing Corto CD also displayed smaller sex combs and a phenotype opposed to that of Corto mutant males who have ectopic sex combs and which could reflect reduced expression of the homeotic gene, sex combs reduced. The Corto chromodomain is a chromatin targeting module. Corto binds polythene chromosomes of the third instar larva salivary glands at many sites. To test the role of Corto chromodomain in chromatin binding, immunostained polythene chromosomes of larvae overexpressing Corto CD in salivary glands with anti-flag antibodies. FH Corto CD bound polythene chromosomes at many sites. Like endogenous Corto, FH Corto CD preferentially bound DAPI interbands and puffs, i.e. regions corresponding to open or actively transcribed chromatin. These results indicate that FH Corto CD mimics Corto binding on polythene chromosomes and that the Corto chromodomain is a genuine chromatin addressing module. These experiments demonstrate that RPL12 and Corto interact at the Corto chromodomain, and it is necessary and sufficient for this interaction. The identification of other RPs among the pulled-down polypeptides suggests that Corto CD interacts with a complex of RPs via a direct interaction with RP12L12.
ETP Corto is partner of Polycomb and Trithorax complexes and participate in epigenetic maintenance of gene expression. Multiple Corto binding sites on polythene and chromosome as well as pleiotropic phenotypes of Corto mutants show Corto transcriptional targets are numerous and involved in many developmental pathways. Previous investigations into Corto partners have highlighted its interaction with several PCG proteins, leading to the conclusion Corto might regulate PRC1 and PRC2 functions. Quasi-systemic presence of RPs at sites of transcription on Drosophila polytene chromosomes, as well as direct interaction between several RPs and histone H1 in transcriptional repression, suggests that RPs could actively participate on transcription modulation. Genome-wide chip-on-chip analysis of RPL7, L11, and L25 in S-Pombe reveal a striking similarity of their binding sites, suggesting that they might bind chromatin as complexes. Results might point to the existence of pseudo-ribosomes composed of several RPs on chromatin. Overall data suggests that pseudo-ribosomal complexes composed of various RPs are associated as on chromatin and could thus participate in transcriptional regulation. Like histones, RPs are subjected to a plethora of post-translational modifications, including ubiquitinylation, phosphorylation, acetylations, and methylation.